under the focus of climate change. And I'm going to kind of go through my work today on mostly our local amphibians. We're very lucky here in Santa Cruz to have a lot of local endemic salamanders and frogs that are actually very endangered. So I'm going to talk about our work on those a little bit and then kind of expand abroad. Yes?
I'm sorry, there is a music broadcast.
Yeah.
Just
Monica?
ask them to turn it off, so I don't know if you want to pause 30 seconds.
Sure, we can pause.
Then. Okay.
Oh, wonderful. Okay, is that better?
Yeah.
Okay, awesome. Good start. Pretty good. All right. So my talk is on the local amphibians here, and they are on the edge of extinction, unfortunately. We're going to kind of go through that. So primarily, we're going to kind of talk about what an amphibian is. So an amphibian is a frog or a salamander, what most people think. It also can be a Sicilian, which we'll go through later, which is a legless amphibian. They interact with their environment, and most of the ones around here in Santa Cruz are actually either endangered or threatened. And what endangered means that their population has significantly reduced in such a way that they might not survive. They might not be able to reproduce and keep populating their environments. Amphibians, kind of the key thing we think of amphibians is they have usually some sort of an aquatic stage to their life. So that can look like this, like a frog reproduction. So we can have frogs, right? I don't know if you've ever seen a frog. They usually live somewhere kind of moist and wet. They lay their eggs primarily in the water. They hatch into tadpoles. So they have an aquatic stage of their life, usually in their larval stage, or some are completely aquatic for their whole lives. And some, like this frog here in this picture, will metamorph and become a terrestrial animal. So amphibians are very interesting because they have kind of like this two-part lifestyle. In the water, on land, some do both. It's kind of an interesting thing. And they are found pretty much everywhere. We are lucky here in Santa Cruz, but the distribution of amphibians is all over the world. There are lots in tropical areas, if you ever think of like a tree frog or a rainforest, kind of like that, there's lots of frogs. There's actually a lot here in Santa Cruz and northern up into California. In my mind at least, when a lot of people think of frogs, they think of things like this. Bright yellow dart frog or a tree frog. We're also here in Santa Cruz where we're not in a rainforest. Our amphibians are not usually as brightly colorful. So we have things like newts, which kind of blend in. I don't know if you've ever seen these walking around. If you go outside after it rains, sometimes they're trolling about, looking around. And we also have animals like this that are actually amphibians. We call them Sicilians. They live underground primarily. They look kind of like snakes or worms. But amphibians have a very large range of what they might look like. And they are ectotherms. So all what we call herps, so reptiles and amphibians, are ectotherms, meaning they are what we call cold-blooded. They are the temperature of their environment. They do not have a metabolism that keeps their body temperature up like we as mammals do. And they must thermoregulate. So they must themselves adjust through their environment, moving different places to get what body temperature they want. Higher or lower, they must move around. Another key part about amphibians is that they have porous skin. We'll kind of go through this later, but all these things make them very strong interactors with their environment. They're very highly affected. Right, us as people, if we go out where it's sunny, we might get hot, but we're fine, or we might get cold, we're fine. Our body temperature stays relatively the same. We stay pretty succinctly at the right temperature. Whereas amphibians or reptiles will become the temperature of their environment. So interacting with their environment is very key for them. This is my little model of how a salamander might interact with their environment. They interact in many ways. They get direct heat from the sun, from bounce back off of ground, rocks, bushes, things like that. They also lose heat through things like wind and convection, also conduction to the ground if they are warmer. And amphibians particularly have evaporation effects with their water, which will change their body temperature. So this is kind of what it looks like when they might thermoregulate in their environment. And now we wonder, why does it matter that we have amphibians and why it matters that we study them? Because they are ectotherms and they interact with their environment so strongly, and because they have life history stages, they're very, very sensitive to changes in their climate and in their habitat. So even slight changes can really heavily affect them because they have these necessities of being in certain temperature environments and also being near water and needing a certain amount of moisture in their skin. We use them as in biology as kind of a signifier of an environmental stressor. So sometimes we see stress in the environment that are occurring that we might not notice as people because our bodies adjust, and some of the first organisms that are affected by these changes in the environment are amphibians. So they're kind of key factors in these environments and are the first ones to show stress when they encounter something different. This means that they are sometimes the first animals affected by climate change. As our climate changes, it can get hotter, colder, drier, moister, and that affects them all very heavily. So they are very susceptible to changes in climate. They are also really heavily threatened by other areas. Some include habitat loss, collection for pets for us as people, and we need to create a rapid conservation plan. You can see that they're declining very heavily, not just from climate change over the long term, but on the short term too, for other reasons. So that's why it's important to study them, and this is kind of what I'm working on in my PhD. What's important to know is what their thermal capacities of amphibians are, so what their thermal ideals are. Something that's called their thermal preference, the temperature they prefer to be at. We'll get to that later. And then we also want to see if amphibians can acclimate their preference, so change what preference they prefer to live at given time to adjustment. And we'll kind of go through all of that. So these goals I'm kind of completing with 3 different species. We're going to focus on the most local one here first, which is the California red-legged frog. Has anybody heard of this frog? Yeah, awesome, great. There's a lot of actually conservation around here right now for them, which is great. So the California red-legged frog is actually our state amphibian, which is pretty awesome, if you didn't know that. They are also vulnerable, so they're not quite endangered, but they're getting very close to being an endangered animal. They are very local. They can be found all over Santa Cruz and Santa Cruz County in fresh ponds. I primarily study them at the UC Santa Cruz Arboretum. There's a big breeding pond in there, which is actually the only breeding pond on the UC Santa Cruz campus for a huge population of frogs. What's very interesting about them and can pose a problem is they are terrestrial animals. They hop around on ground, but they need water to live and breathe in. So they actually do this weird thing where they migrate. So these frogs are migratory. They will migrate to a breeding pond, stay there, breed, lay their eggs, and then migrate out. So they have to traverse, I guess, a large territory. They're moving back and forth a lot, and they need these great breeding ponds. The reason they're vulnerable, there's many reasons, habitat loss, collection, stuff like that. But there's 2 big ones, which are invasive species, primarily with the bullfrog, and this fungus called the chytrid fungus, which we'll get to later, that's affecting some of our amphibians. So at the Arboretum, this is what the pond looks like. It's not the most beautiful of areas, but the frogs really like it. It is full of nice coverage and debris, which they actually really like, which is good for them. They like places to hide, places to move, all that kind of good stuff. So at the Arboretum, it's actually really ideal. So we talked about those 2 big issues of bullfrogs and chytrid disease. This pond is great because it actually dries up each year. So the frogs come during their heavy rains, they lay their eggs, they breed, they leave. Their eggs hatch, become tadpoles, the tadpoles eventually metamorph, and then the pond dries up completely in the summertime and disappears. Which is great because that means bullfrogs can't thrive there, they need water year long, invasive fish can't thrive there, nothing can really live there that really harms the frogs traditionally. And it's thought to be a chytrid-free area, so it's free of this fungus that's really heavily affecting them. Long time ago, in the 80s and 90s, until the 90s actually, Arboretum actually thinned this pond. So they thinned the canopy, they thinned the overstory, they trimmed everything for aesthetic reasons, for us as people, so we could go and enjoy the pond. But the frogs actually liked it that way too. Once the frog was discovered to really be there and became vulnerable by our state government, one of the kind of policies, I guess, is when you see an endangered or threatened animal, it's kind of a hands-off policy. You want to leave them alone, you don't want to disturb their environment, so all thinning and cleaning up of this kind of overstory and this canopy stopped. And since then, it's just become really overgrown. If you go out there now, you kind of almost have to use a machete to get to and from the pond. It's not pleasant. And we can't even do the same surveys that we used to do at this pond to see how it goes. There was a study done a couple of years ago to see how the conditions were for these frogs. And it wasn't great. It's become very cold there, and they don't like that. But very, very conveniently, last winter, so 2 winters ago, I guess, a huge storm knocked out a huge amount of trees in one specific area, so much so that the Arboretum had no choice but to clean it up with permission from the government. And they kind of got back to their own condition in this one section of the pond, the old condition that was thin and ideal for frogs. So we've been going out there and wanting to know what the new thermal capacities of that environment is. see how well these frogs can thermoregulate, so can interact with their environment, can get to the temperatures that they do or do not want to be at, and survive in those conditions. And there's something called microclimates that we want to find. Normally, I think when a lot of people think of climates, they think of the big, large, kind of citywide, countrywide, statewide, anything like that, big climates. We have something we call microclimates, where in a specific area, there are very different climates. So in the sun versus the shade, under a tree bark, not on a tree bark, slightly in water, not in water. All these slightly different conditions actually have quite a bit of temperature difference. If you're a frog that's like this big, it's very easy for you to move into those different microclimates and adjust. So we want to see what those microclimates do. So ideally, we'd go out, back to this model, put a frog or a salamander out in these different conditions and measure how they react. That would be ideal, but we don't want to go out and tie endangered frogs and amphibians to rocks out in the middle environment. For their safety and for us as biologists, we want to preserve them. So we do this kind of interesting thing. We make a model. That's kind of what it looks like. Looks like a little frog in this case. I don't know if you can see it. It's made of agar, which is a sugar compound. So we make these kind of, my undergraduate volunteers call them jelly frogs. They're little like jelly. They're made of a sugar compound that you heat and add water to, and they dehydrate like a frog might. They lose water like a frog might. So we can use these to kind of represent what a frog would do in a normal condition. So what we do is we go out and use these agar models to represent real frogs. We put what we call a hobo into a model. So hobos are these temperature devices. I meant to bring one. I'm sorry, I don't have one with me. But they are temperature loggers that take data. So they'll take a temperature reading every minute on the minute for a period of about 2 weeks. So each of these frogs has a little hole in the side. You plug in one of these thermocouples, is what we call it, like a thermal logger into these frogs. And we divide the pond into kind of 8 research slices. And we're going to put these models out at these sites and kind of measure the different microclimates in different areas. At each of these sites, we kind of try and create these microclimates. So we put out models in the sun, in the shade, in wet environments, and dry environments, all the different kind of combinations of those to see what their different thermoregulatory capacities are at different microclimates. We weigh these models over a period of time to measure water loss, which is which kind of represents the way an amphibian would dehydrate. We also measure the temperature. So using those two, we can mathematically create the thermoregulatory capacities of these frogs in the environment. And we also measure the air temperature, the humidity temperature, the relative humidity, to see how they're interacting with just the air too as well. We do this, we have to, go to the next slide, deploy these models for the whole season. So also seasonally, the microclimates change. So in the winter, when there's a lot of rain, or in the summer, the microclimates are changing. So we have done this over every season that we can. The frogs are out there. We analyze that data for temperature and dehydration rate. It's kind of a picture over here of what they kind of look like. It's hard to see in this light, but put these little frogs attached to a hobo that have plugs in them, in the sun and in the water. So these are both sun models. We put these cages over them. The birds like to eat them. Apparently they represent frogs very well. And the birds think they're food. Which is good. So we are going to use these results to recommend conservation for these frogs. So right now the Arboretum does have plans to trim more of these trees, which we can kind of show here. I'm jumping ahead. But when we go out there, this is kind of what a temperature logger looks like when you analyze the data. It has this big ups and downs of temperature that we're going to analyze and kind of find the averages, the means, the different quartiles of what the temperature looks like at different times of day. We also have been going out and measuring the frogs. We're really lucky and fortunate to have a lot of colleagues that have permits to work with endangered animals. So the UC Reserve System has been helping us a lot with the collection of these frogs. We weigh them, we measure them. Kind of looks like this. That's one of the really big adult males. They're very chunky and very unhappy when you catch them. And there's me. We're doing a lot of different processing. We are trying to measure them, weigh them, also see what their body temperature actually is to make sure our models are correlating well with the real animals. And we also do chytrid swabs, so we're swabbing for different kinds of fungus. Something else that we do to make sure that our models are working is we actually take the temperature of the live frogs, which sounds easy, but it's not because they don't like people and so they jump away from you really fast. So you have to measure the temperature from very far away. We use this thing we call a temperature gun. It's actually really cool. It looks like a gun. It shoots out a laser, and whatever that laser hits, you get the temperature of that animal. One of the hardest things is finding the frogs. So we were very fortunate that this year a couple of these large frogs had a whole bunch of babies. So we went out there one day, and there were just hundreds of little, we call them froglets, little mini frogs, out there. I don't know if you can see, this picture is actually only 4 feet away. If you can find a frog, I can't. Then you get like 2 feet away, you still can't really see the frog. Eventually, it's kind of what a little baby looks like. And it's very blurry because that's about as close as you can get before they hop away from you. But they're cute little frogs. They have this nice kind of brown color. They blend into the ground, and they don't really get this red leg, as we call it, from the red-legged frog until they sexually mature. And they hide up really well. So with all this data that we've collected, both from the models and from the live animals, we did get some preliminary results. In the one slice of this pond, this one experimental site where they did thin the trees because of the storm, they did find that there were higher thermoregulation capabilities, which means that they have a bigger range of thermal environments to choose from. Better way to thermoregulate. They can get warmer. Why does warmer matter? They need to get to certain temperatures to be able to breed, find food, to reproduce. Also to fight off this fungus that we've been talking about. So that's all kinds of great stuff. So because of that, we're supporting the thinning. The Arboretum is currently raising funds to thin more of this pond, which is great. They're looking for money. Unfortunately, we did find chytrid in this pond, which is a not great thing. So this pond does have chytrid, or at least some individuals with chytrid fungus. So they're working very hard to thin these trees and find funds to thin these trees. And using that and the data we collect, we hope to thin the trees and then redo this surveying to see how they are expanding and working on all of those. So why this matters in the long run, because that is one very small specific pond in our county, is it will work for our larger county. We are working with other local, they call themselves frog experts, I guess, frog conservation people that are working on that for private companies or for academia matters, like for our school or university, to find these thermal capacities and make sure that the ponds are at that thermal capacity that they can reach. So this kind of study was all about the environment and how the frogs are interacting with their environment. What's also really important is to understand what our amphibians actually want. So this study is what we think they want, pretty much, and what the pond does for them. We want to know actually what these amphibians need to be at for their thermal preference. And this is also just one location, and we want to expand, more broadly and see how they might encounter for the whole state, the whole country, and also for animals that migrate, we need to include not just their breeding habitat and their breeding ponds, but the larger environment that they interact with, so the different environments they might move through as they migrate to their adult life area. So the way we're going to do that actually is with another local amphibian, and this is actually even more local than the red-legged frog, which actually is throughout California. We actually have a species, it's called the Santa Cruz long-toed salamander, and they are endemic solely to Aptos. They live in about anywhere between 6 to 10 ponds, depending on how the weather is, and they are awesome, but very, very endangered. They are only local. The freeway is built right between all the breeding ponds, so that hasn't done very well for them. But they're pretty awesome, and we are actually very lucky to work with them through more collaboration with a conservation company that actually is being hired by the government to study them out in Aptos. So they migrate as well, just like the frogs, and they migrate through many different habitats. They migrate through things like willow, through oaks, through shrubland, through grassland. They interact with a lot of different temperatures, and they're thermoregulating through all these different environments as they move. So another great picture of what they look like. It's really hard to find good pictures of what they look like. They're pretty endangered, but they're cute little black salamanders that tend to have orange or gold sparkling on their backs. They're about this big, not too large. Where they're found, picture is slightly small, but right here we've got the one, and just these kind of 6 ponds. And as you can see, unfortunately, they're all surrounded by housing developments, and these animals must migrate pond to pond between all these houses, which is another struggle that they are trying to get through. So on this study, we're particularly focusing on the different habitat types, so less so on one specific breeding pond, but the thermal capacities of different environments that these animals might go through. So that includes the things we're talking about, like willow, oak, grassland, and measuring those thermal capacities of those areas. This is kind of what it looks like over there. It's kind of hard to tell, but on the right, there's grasslands. Left, there's more oak. As you go farther down, they approach even some willows. And so they're interacting with a lot of different environments. This is closer to the pond itself. It's kind of hard to tell, but the pond is, this is the pond. It's just covered in reeds. So we do very similar things where we put out these agar models. Instead of looking like frogs, they are cylinders. The salamander is relatively a cylinder. We do a similar setup where we're putting these agar models out at different microclimates: sun, wet, shade, dry, and the different kinds of environment that they have. But we're also doing something new. We're hopefully going to do it at the Arboretum as well. So we're putting out these really funny little loggers. They're called tidbits. They take temperature data of the water for a whole year. So we've been putting these out at local ponds all over the place. You hammer in a piece of rebar with this tidbit on it, and it will collect our local temperature data to see how it fluctuates year to year as the environment changes with the breeding. The breeding ponds changes with the environment. We are also really lucky to add on to this experiment. We were able to get some of these animals into labs, which is not easy to do when they're highly endangered. But we brought these 10 animals into lab to measure their thermal preference. We'll get to what thermal preference is, but when we bring an animal into lab, we do all the kind of normal things like weighing it, measuring, taking pictures to keep for our records. We also have been swabbing for chytrid. So chytrid is this fungus that is very harmful to amphibians, and we've been pretty much swabbing every amphibian we find ever locally to see where it might be prevalent in our community, see which ponds may or may not be infected. So we've been swabbing for chytrid, and testing for thermal preference. So what is thermal preference? It's kind of what it sounds like. It's the temperature that these animals prefer to live at. This temperature is their kind of ideal body temperature, where they have, it optimizes their function, it optimizes their reproductive success, it optimizes their survival. It's their temperature that gives them the best success rate. It's kind of what this is a thermal preference curve. The body temperature is on the bottom, and performance can be measured in a different way, but how well they are doing. And you can see that this animal, this make-believe animal, has optimum temperature right here. And at that temperature, they'd have the best rate of survival, the best rate of reproduction, which is what they're striving for as animals. So we want to see what temperature they prefer and see if that actually matches their environment. So we do that in this actually very cool way. We make a track. That's what we call it, kind of like a track. It is this arena where there's a thermal gradient. So one side is very hot, one side is very cold, and there's a gradient in between of all the temperatures. And the animal is freely allowed to move in this track wherever it chooses. So it's very low impact on the animals themselves. They are just kind of doing what they do, and they walk around, they thermoregulate, which is what they do naturally, and we can measure what temperature they prefer to thermoregulate at. And to do that, we measure their body temperature every 10 minutes in this track for 2 hours or 3 hours, depending on how it's going. These tracks look like this. They're very sophisticated, but they're really just boxes, very long boxes. One end has a nice piece of heat tape, one end over here has an ice pack, and we get this nice thermal gradient. And then, just like in the frogs, we measure the actual body temperature of the salamander with a thermal heat gun. So we're just measuring what their body temperature is at. And because they are, as we call them, ectotherms, they are the temperature of the environment. So they choose to be at a temperature, then their body is that temperature. So you can see there's a little tiny salamander right here, kind of crawling towards the cold end of this track. Using this data, we get the thermal preference, which actually is very important because then we can compare that to these agar models and these other data that we've been collecting locally at the ponds to see if they correlate. We can also use this model, this data to make a model. So my advisor particularly makes models under how animals will fare under extinction for climate change. So we know pretty well how our area will do locally under different kinds of climate change. And we can use this kind of data to see how, like, the long-toed salamander or the California red-legged frog will do under certain conditions for climate change, how they will kind of fare in their environment. Something that's really cool about the salamander and kind of where I'm expanding to next is that the genus that it's in, so kind of the level above species, the genus that it's in, the Ambystoma genus, is actually very kind of a cool genus to talk about. It ranges through pretty much western U.S. into the mid-U.S., kind of towards Nebraska. and then down into Mexico. What's very interesting about this genus is it actually has a lot of different what we call life history strategies. So life history strategy is just the kind of life path an animal goes through. And so we talked about earlier, a frog lays its eggs, become a tadpole, they metamorph and become a terrestrial animal. That is a life history strategy with salamander. There are a lot of salamanders, like the long-toed, that do that. Like a lot of salamanders that you probably think of. They're a terrestrial animal. They walk around on land. They go to ponds. They lay their eggs. Their eggs hatch into little larvae. The larvae swim around in the ponds for a while. They grow. Eventually, they metamorph and become a terrestrial salamander. That's only one kind of life history stage. Salamanders can have quite a few, and this particular genus that the long-toed salamander is in has all three. So another kind of life history strategy that's pretty cool, and the animal we're going to talk about next, is they maintain their aquatic stage for their whole life. So they are born as a little larva that swims around, and they actually sexually mature to become an adult underwater. They're what we call a pediomorph. They maintain their larval characteristics, and they live underwater for their whole life. So a species might be a terrestrial animal, a species might be a sexually mature adult as an aquatic animal. There's also other species that can choose their life paths, and those ones are also very interesting. So depending on what their environment is at, they could choose to become sexually mature as an aquatic animal or metamorph into a terrestrial animal depending on what might fit them best in that current environment. So the plan is to study the whole genus that the Santa Cruz long-toed salamander is in. We're starting that now. We're planning on putting out agar models at ponds for all this genus. There is approximately 20 plus animals in this genus. There's a lot. And we want to put in these year-long data loggers, agar models at all these ponds, measure the temperature preference of these animals like we did with the long-toed salamanders. And we also want to do one more kind of important aspect. It's called acclimation. We want to see if they can acclimate. So what is acclimation and why it really matters? So acclimation is the animal can physiologically adjust to a change in environment, and particularly in our case in temperature. They can adjust to increase almost the preferred temperature. So I have this graph. So it's just kind of the curve, maybe a preferred body temperature of an animal. Its preferred optimum temperature is here. If their environment gets hotter, like it might under climate change, this could happen where their body temperature increases and it's no longer at its optimum. And perhaps now they don't do very well because it's too hot for them, they're not longer at their optimums, they're not thriving as well. Or there's this possibility that if they are young and what we call a plastic animal, they have the ability to adjust their preferences, they could actually overall increase their thermal preference higher. So they are increasing their optimum temperature along as the climate is moving. They are moving with the climate. The way I like to think about that is surviving versus thriving. So I'll actually go back. In this situation, anywhere in this curve, the animal will live. So at this higher body temperature, the animal still lives. It will live. It will survive. But at this optimum temperature, it thrives. It has the best chances for success. It has the best reproduction. It will thrive. So that's what we want to see happening. It's also important because in these three life history stages that we just talked about, different animals might be differently plastic, is kind of what we're talking about. Salamanders that do not become a terrestrial animal, become fully aquatic, live in a lake or a pond, and they are stuck in that lake or pond. A lot of times when we're making models for how animals will fare under extinction, we assume some amount of the animals will migrate either to parts of the range that are cooler, or they'll extend the range into cooler regions, things like that. Animals that live in lakes and ponds they can't do that. They are stuck where they are. They can't just get out and pick a better pond. So it's really important that they maybe have this ability to acclimate to fight off the climate change that are occurring. We kind of call that the rescue effect. They can't rescue themselves. So the species that we're focusing on mostly for that is what we call the axolotl. It's the Mexican axolotl. They're these really cool animals that are native to Mexico. They are fully aquatic. They live their whole lives with this cute little dark face. They do look like that in real life. They are critically endangered, very hard to find, and they do spend their whole lives as a pediomorph, as an aquatic adult. So this is what an adult looks like. So they're forever underwater. They only live in a couple lakes, and when they're there, that's where they are. They're stuck there. So we don't want to go out and take critically endangered animals out of the ecosystem. That's bad. That's against what we want to do. There's actually a lab on the East Coast that breeds axolotls out of the wild strain. So we have a whole bunch of axolotls from there. What we want to do is test for their acclimation potential to see if they can actually raise their thermal preference. So how we do that is we take a whole bunch of larvae. This is what a little baby larva axolotl looks like. They're like an inch big. They're very cute. And we raise them in different temperature conditions. So we're raising them at 15 degrees, which is where the cooler part of the environment is. 2 degrees higher, which is the moderate prediction for how climate change might affect their environment, and then 21 degrees, which is the highest effect that their climate change might happen and occur in their environment. We do know that animals can survive in all these climates, but what we really want to know is if they can thrive and if they can change their thermal preference. So like the long-toed salamanders, we have to make a kind of like a arena, a track, to measure their thermal preference. It becomes slightly more difficult because these animals are fully aquatic, and if any of you know how to make a thermally aquatic gradient, help would be great. We're almost there though. But this is what it will look like. We'll have this kind of outside trough, this wood trough, that's separated into 3 chambers. One chamber will be heated with water, and one chamber will be cooled with water over here. The actual axolotl itself will be within another chamber that sits within this to heat its water. We don't want to directly heat the water that the animal is in, and this kind of metal sheet that this animal is in will be cooled on this side, heated on this side, and will create a thermal gradient. And then, as per the long-toed salamanders, we'll just place the little larva in there, and they can happily swim around and pick which temperature they prefer to live at, and then we can measure that temperature. Using that, we can actually see if their temperature preference changes. We hope that as their temperature rises, they've been born in higher and higher temperatures, that their thermal preference would actually increase. Larval animals tend to be much more plastic than adults, just like human kids are much more able to adjust than human adults are. They are able to thrive. So we're hoping that in this larval stage that they can actually change their thermal preference and increase it if they are in these hotter environments that will cue their body to increase their thermal preference, and they might be able to physiologically adjust. So that's kind of what this picture looks like. We're hoping that their thermal preference will move. It will increase as they go. And we do predict that this will happen. We also predict that younger larvae will be more plastic, more able to change than older and adults. We've already seen that the adults are much less able to handle changes in temperature than the larvae have, just from housing them in the lab. The goal is to see how this also changes between the different life history strategies. So these animals have always been stuck in the water. They might not have that much ability to acclimate, whereas species that can become metamorphs or can choose either life path might be more plastic, more ability to change. So the plan is to see this across the whole genus. So we're planning on trying to bring every one of these genus into lab, raising larvae, seeing the difference in their acclimation potential, their ability to change their thermal preference. We also want to see, there's a very good chance that there's something in thermal acclimation that we call maternal effects. So in a lot of animals, and reptiles too, moms that interact with their environment can pre-program their offspring to handle differences in the environment. So if a mom is raised in a higher temperature environment, even if it's hard for her to survive in, she knows it's hot, and she can actually pre-program her offspring to already have a higher thermal preference than maybe she does, or they would naturally without this kind of cue. We want to see that exists, which requires raising the larvae we have in that house picture, the little babies to adults, spending their whole lives in these higher temperatures, and then seeing if their babies have even higher thermal preferences. We're going to put agar models out across Mexico and all of Western United States. A lot of my colleagues actually are here who are going to help us with that, and hopefully get this data across the whole Western seaboard pretty much, which will be a lot of work, but will hopefully help us create a well-rounded picture of what will happen. A lot of models, I think, focus on primarily one species, which are great, but we have a lot of species that are going to be threatened by climate change, so thinking on the larger picture side of things. We are going to measure the thermal preference for all these animals, so the temperature that they prefer to live at. And then we are also going to try and get the acclimation potential for the genus. So right now we are just starting with the axolotl. They are easy to get and house in lab, and they are doing pretty well. And then the plan is to also get a lot of our other local salamander species that can be brought into the lab and see if their potential can be raised. And then also abroad. We have a lot of colleagues abroad that are going to help us do this in these labs, and we can collaborate and collect it all together. So the rest of the genus looks like this. We all love salamander species. There's just a couple of them, but they're all adorable, I think. And so we're going to get to work with all of these. We have some that are local, like this guy, which is the tiger salamander, which we have our native California tiger salamander that's also found in Aptos and Santa Cruz. And we have everything from the axolotl, which we have now, to like the marble salamander, all these different animals, which are all part of the same genus, but actually very different and very distinct. And so collecting them all, although they're closely related, will give us a good picture of how maybe salamanders around the whole kind of world interact. It kind of gives us a great summary of an entire kind of group of animals. So the purpose of this is to collect the whole genus, and then use this to create an extinction model. Using this data, we can create a model of how these animals will behave and interact with the climate as it rises and increases, and see how they do. And areas that they particularly need help in, areas that they might not need help in, places that would be the best place for us to put our effort into helping them. So the plan is to eventually turn this into a conservation plan to help with their movements, both locally and abroad. So we're already starting kind of at the local level here with the California red-legged frog and the long-toed salamander, but hopefully kind of abroad and do a larger scale picture, especially for these animals that migrate pretty far. So I'm very lucky that I get to work with a whole bunch of people on a large-scale project, and the information we're going to collect is going to have implications here locally and abroad. So we are currently working on the conservation plan with the Arboretum. We've been talking about it. We now have permission to thin the pond, which is great, and that was hard to get to begin with. But now we actually have to raise the funds to thin it, and they're going to thin certain sections at a time and see how the pond goes, and we're going to keep creating analysis of that and see how the frogs are faring, see how well they're doing. We're also trying to working on chytrid testing across our state into the different areas. Unfortunately, which is expensive, but we're working on that too, to see where this chytrid is, primarily focusing on our amphibians, trying and help those in that area. The Santa Cruz long-toed salamander, as we said, is surrounded by housing, and its breeding ponds are crossed through by a freeway, so it's struggling very hard. And they currently have a very, very strong conservation effort on them to count their population. If you go out to their breeding ponds, they literally, every time it rains, they recount the entire Santa Cruz long-toed salamander population to try and keep it up. But what we're hoping to do is show where there are promising areas in their habitat, which we need to help preserve, and areas that are not, especially with the housing that's being developed, and they are trying to kind of not expand, but thin different areas of habitat for human use, and trying to see where that might be helpful to frogs or hurt the salamanders, and try and help create boost their population levels, help them out where we can. The large-scale amphibian models, we're working with a couple labs across the country. There's a large one in Mexico, which the advisor's actually here, which is great. And we're collaborating internationally and trying to create a conservation plan for amphibian groups across different governmental bodies, which is not always easy for these animals. Yeah, almost done. I have lots of people to acknowledge. This project that's this big has a whole lot of collaborators. The only way to do it, my lab, all my undergraduate volunteers, Arboretum, the UC Reserve Systems, UNOM, different environmental consulting companies who've been letting us use their permits to kind of interact with these animals. Have all been great. And then I have time for questions. If you've never seen an axolotl, they're apparently very popular, and there's all kinds of little gifs and quotes and puns based off axolotls. So I have a few for you to enjoy if you want to ask me some questions. All right, thank you. Yes.
What's the name of the fungus again?
The fungus is called the chytrid fungus.
And is anything being done to try to control that in some of these habitats?
The problem is that it is hard to control. Once a pond is infected, it's not just the amphibians that have it; it's in the pond itself. So it's primarily important not to get there in the first place. So we don't want to spread chytrid from one area to another. That's the key thing, is preventing chytrid from spreading in the first place. There are some methods of not cures but treatments for the animals, but they're not very successful because you have to treat individual animals and not the pond as a whole. You can't go out and clean out an entire pond that has a whole different kind of biodiversity of organisms. But they're trying to work on testing it and then finding better testing methods. The UC Reserve System is trying to create a better way to test environments for chytrid, not just animals themselves. Testing animals is great, but you have to swab each animal. Swabs are not easy to get. You have to run every swab. Not every animal in an environment might be affected. So they're working on a more efficient way to do that. And then hopefully we'll be able to use that to kind of see where that chytrid is throughout our community. Yeah.
What's the population of red-legged frogs at the Arboretum like?
Well, this year we went out a couple times during the breeding season, which is when you're going to find the adults at this pond. We were only able to find, I think, a total of 5, which is not promising. No. It is possible that we missed some, but we went out with one of the UC Reserve directors, who is the one who monitors them, and that's all we could find. We are lucky that during the hatching season, so they hatched really late because of the late rains, the heavy rains last year. So I think it wasn't until August that the tadpoles actually metamorphed into little fraglets. During that day, though, we did find hundreds, but the herps don't usually have a tendency for all the little babies to survive to adulthood, and they do have to migrate up. The red-legged frogs migrate up to the northern part of the UC Santa Cruz campus. That's where they live when it's not breeding season. So they have to survive. They have to survive the migration. They have to survive the migration back. So we're hoping that increasing the thinning will help their population grow. Yeah. Mm-hmm.
So it's very interesting that you are trying to find the optimum temperature that the creature prefers in your
Yeah.
Temperatures in the environment vary by season.
Yes.
So they're colder in the winter and warmer in the summer, and we have fog and so on. But the other thing that varies by season is light.
Correct.
So how are you controlling for dark light exposure in the lab to help them know what season they're supposed to be in?
Well, in the environment, when we're actually outside doing the data, every season we collect actually, there's not, I don't know the scientific word, we call them foliage pictures. We literally take pictures of, at the salamander level, what area above them is percentage of sun versus a tree or a bush or something. So we literally take a picture here, look up, and then count the little pixels of what is sun shape. In the environment, we're trying to take it for the seasonally, so we are trying to get animals all the season. Luckily, things like the axolotls that we have programmed in lab, that live in lab, we actually have them in what we call a controlled environmental room, which is great. They're in rooms that actually maintain their environmental temperature all the time, so we can kind of modify that as necessary. But for the in-lab, we've been taking animals for the thermal preference during that time of year. So like the Santa Cruz long-toed salamanders we got in the winter, we'll hopefully get some again later this spring. They're only fortunately able to get them usually during the breeding season when they're migrating pretty thoroughly. If not, they normally hide pretty well. We are trying to accomplish that. With the models themselves, we are putting them out every season possible. So the agar models will be out during every season. The tidbits, the little temperature loggers, stay out all year. So we are trying to account for that.
In the lab, do you change the length of the nighttime?
So a lot of these animals, like the Santa Cruz long-toed salamanders, actually only come in during the day. So they're in our lab for less than 4 hours to prevent any possible upsetness, I guess. Upsetness is not a word, but irritation to their system. So like the Santa Cruz long-toed salamanders were brought to us at 12, and they went home at 3:30.
And then where do they go home?
They get put back in their natural environment. So we try not to bring animals into the lab as much as possible for the long term. We're trying to work with our local populations as much as possible because they are endangered. We don't want to actually keep them in lab on the long term. So they're only coming in for short periods of time, and then they get put back into their natural environment, and we're hoping to do that a couple times throughout different seasons to get their environment. Yeah.
What is the lifespan of salamanders?
Salamanders actually live a crazy time. I think a lot of people don't realize that most reptiles and amphibians actually can live a long time. So axolotls, long-toed salamanders, a lot of these ones in captivity can live 20 or 25 years.
Wow.
Yeah. In the wild, obviously their chances are not that high. If they live to adulthood, they can still live to be 10 or 15. I know they've been finding Santa Cruz long-toed salamanders. They've been trying to track them that are at least 8 years old, but it's kind of hard to keep track of individual salamanders. Yeah.
Do you actually eat?
So I was talking to the long-toed salamander guys. I guess you can take a small sample of one of their fingers and send their bones off to analysis, and they can count rings in a bone like they can rings in a tree, calculate how old salamander is. So that's what they were doing to estimate, but it is still just an estimation, because salamanders are pretty awesome. When they lose their toes or their legs or anything like that, they regrow. So you don't happen to know that if you have how old your salamander is by just taking its toe. But that's kind of how they've been estimating. Most of our averages are from captivity because we can keep track. But yeah.
So you said that salamanders can choose to morph into being a or
Some species can. Sorry. Some species.
So do you find that like in a particular environment that the majority of them would choose to either be aquatic or terrestrial?
I'm still looking kind of into
Regina,
that.
you might need to repeat the question.
Yes, repeat the question. For salamander species that have the choice of becoming aquatic or terrestrial, so they can choose, do they tend to all choose one way or the other in a certain kind of, I guess, grouping or
In a certain environment.
To be honest, I don't know that much about it. A lot of ponds where there's a possibility of the pond drying up, yes. So if you live in a pond that decides to dry up on you, you have no choice but to metamorph into a terrestrial animal. But it can depend on so many different things. It can depend highly on temperature, on food abundance, on the amount of competition you have for food, so how many siblings you might have in the pond, the different kind of predation that you face, there's disease. Some salamanders tend to be cannibalistic, so how many of your salamander siblings have become cannibalistic? There's so many factors that can play a role into which choice they make. Yeah. Yeah.
So you mentioned that herpetologists study amphibians and reptiles.
Correct.
Anything else?
I believe that's it, right? Am I missing something? No. No. Yeah.
Question over here.
Yeah.
Are any of the frogs or salamanders dangerous or poisonous?
We do have one local amphibian called the California newt that is very highly toxic. I think the estimate was one newt can kill 10 people, so I wouldn't go around eating salamanders.
And what color is that?
It is brown and orange. They are still safe to hold. They're not going to infect you or bite you. You would have to actually consume them to be intoxicated. So as long as you don't go around eating salamanders, you should be okay. And wash your hands, yes. In the back.
Is anybody looking at our local giant salamander?
Yes, we're trying. They are working on I don't know if you've ever heard of the FERP. It's called, what's it called? The Forest
Ecology research plot.
Forest Ecology Research Plot that UC Santa Cruz has been monitoring, and they have been trying to keep track of the population up there. They just sent me a couple pictures the other day, actually, of some really cool ones. My lab personally goes out to look for them all the time, but they are not the easiest to find here locally. They're not in high abundance. But they have been monitoring up there on the FERP. But that's about as much as I know on the giant salamanders.
So earlier you showed like a slide of the ponds like kind of broken up by the residential area.
Mm-hmm.
Are you finding that there's like less genetic diversity like that in ponds?
So that's the problem with the ponds
Can you
is,
repeat the question?
yeah, so for the long-toed salamanders, where their ponds are very separated geographically by housing developments, if ones that are farther off are less genetically diverse than others. I do not know the exact answer to that, but the biggest problem that they're having is they do need to migrate between the ponds. So that's being the biggest issue, is that animals are just not surviving migrating. You should see some of the like historical migration paths literally have like giant hill where the animals climb up, a house, a road, a house, and they have to somehow manage to get to the other side. It's not like a small effort. They've been trying really hard to build these underground tunnels between one hill and another, but then you have to get the salamanders to use the tunnels, which doesn't always happen. So they're trying very, very hard. but it's not ideal for sure.
I send it to my work because I work for the district.
Yeah.
But they didn't
So
start the first
I've been asking
Wednesday or
questions.
the last
Yeah.
Wednesday. You see, I can handle that. Okay, cool. This is again. No,
What's so
everybody's
special?
on.
Why are the salamanders?
Science on tap.
I don't know much about it. So originally it was thought that these long-toed salamanders were a subspecies of another long-toed salamanders. But lots of people, including my advisor, would argue that it's its own species, and I think they're kind of working on defining it that way. Why it's here locally, I do not know. I don't know, does anybody at this table know? I don't know. Carla, do you know the answer to that question?
I guess would be competition.
Yeah. The fog, maybe. I don't know. We're actually very lucky. I don't know if you've ever just gone out on a rainy day. Santa Cruz has a very high hotspot for salamander diversity. There are lots and lots of salamander species here locally. Much more than you might think if you just kind of went out and looked for them. And Carla has mentioned there is competition between species. So each species has its own kind of what we call like a niche, its own kind of area that it thrives and works in. And they try and differ between animals. So as soon as niches overlap, you get competition. So they kind of diversify to have different kinds of environments that they fill. So it's possible that they are here because of competition, that they can't expand out because they're locally. Most salamanders are also restricted to areas that have water. So they can't just transfer 10 miles away without having a pond to get to. So they don't migrate very far. When we're talking about this picture, I don't know. Let's see if I can find it again. This picture's not very large. From here to here, to each side of the picture, you can't see that. Sorry. From here to here is one freeway entrance. So these entire species are living between two freeway exits. It's not very far apart. Yeah.
There are a few other spots for that.
Yes, there are.
Farther away.
There are some original ponds that are across the freeway. Obviously, they don't really go across the freeway very well anymore.
They're down in North Monterey County.
Yes, but I can't speak to those populations because I've not seen them. But
Well, I just want everybody to know they're
Yeah.
really endemic to this spot.
Yeah.
They are a few other places, so they've gotten around. 10,000 years.
yeah, before we were here making freeways. Yeah,
In those drought years, which,
yeah.
drought years, was there a significant percentage decline?
So from what I've only been studying this population for about a year now, but I've been talking with the conservation company that's working here. Even this year, we kind of had that delay in rains for a couple weeks in the middle. Some of their ponds that normally don't dry completely dried. And they moved, they migrated a lot less. So these species that live a long time don't have to breed every year. So on really bad years, they might not. So this year they noticed a much lower number of animals moving between ponds. And they're predicting it is because the delay of rains dried up some of these ponds. That's just their kind of hypothesis on what's happening. Yeah. Yeah, last one, go for it.
I have two questions.
Go for it.
The layperson question, the question that had to do.
You can do both, that's fine.
Is that okay?
Yeah.
So breeding ponds, once they breed, they need to move to a different pond?
Some animals move between ponds, some don't. Some move into, not dry habitat, these habitats are still moist, but away from ponds into different areas. So like the red-legged frogs move northern, kind of under redwoods, where it stays moist most of the year, but there are no physical bodies of water necessarily where they're moving to. Same with these guys here. Yeah, go for it.
Just the other question had to do with the bullfrogs,
Yeah.
right? And then you introduced this idea of
Maternal effects, yeah.
Yeah, so how do you know if the bullfrogs that don't have effects?
They could, totally. Bullfrogs are not native. And unfortunately, they're one of the biggest threats to our amphibians and a lot of native fish species because they're very aggressive and very hungry, and they'll eat anything. They are not picky eaters. I don't know much about bullfrogs myself. The nice thing about bullfrogs is their larvae have to live a whole year in water before they can metamorph, or it's a year and a half, something like that. So a lot of these ponds that dry locally, that our amphibians are adapted to, they migrate away when they dry up. These bullfrogs don't, and then their offspring all die, which is why these dried up ponds are great. Our amphibians are used to them, and they have adaptations to them, and the bullfrogs don't. And so that's kind of why we like these ponds that dry up. They work well for our amphibians and not so much the bullfrogs.
Any last burning questions?
One in the back.
Have these animals had their genomes sequenced?
The axolotl has. They just did the entire axolotl genus a couple of weeks ago, actually, which is really cool. It is very, 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 very long. People like the axolotl particularly because they have great regrowth of body parts, including like brains and organs and stuff. So they're used a lot for like cancer research and other kinds of stem cell research because they can regrow almost anything. You can watch an axolotl like regrow its arm. It just does it. It doesn't have any qualms about doing it. It just goes for it. And they can even, I think they found one that's regrowed half its brain. I don't know. It's a cool animal. So it has had its whole genome. The Santa Cruz longtoad has not. I do not know about the red-legged frog. My guess is no. If not, I feel like I'd know about it. But yeah, I can always ask questions after too. I'm happy to.
If there's another one, go ahead.
Go for it. Not that I know of. Carla would know that. That's her study organism. We have another salamander. It's called the California slender salamander. They're very slender and petite.
They're the dachshund of salamanders.
Yes. The question was if they are endangered or threatened in any way.
They're not listed as anything. They're a species of least concern. However, no one has done any population studies counting the number of them that are in the environment since basically the 1920s. And that was one study in a small plot up in Berkeley where someone put down a board, removed all the salamanders under the board weekly for, 7 months or something. And from that one study, they estimated it's something like 14,000 salamanders per acre or square kilometer, which are two totally different sizes. But that was in the 1920s. That was before some climate has been warming, pesticides have been introduced to the environment. It was one study in one area. Nothing modern has been, or more modern than that, has been done, and nothing in any other parts of California that are comparable, except there's been another study in like Angel Island, but that was an introduced population. That was a study done in the 50s. So again, very little known about this. They're still pretty commonly found, but anecdotally, the populations are probably decreasing. If you talk to the expert on California salamanders, Dave Wig at the Museum of Vertebrate Zoology up in Berkeley, who's been catching them for decades, they seem to be in smaller numbers, but there's really no formal data.
What do they eat?
Salamanders eat any kind of insect and vertebrates. Some of the big guys eat other salamanders and amphibians. They'll eat fish. Larval stages will eat more, sometimes vegetation, but they're primarily kind of carnivores. They're not very picky creatures, though.
On that note,
I hope that you all learned a little bit of something tonight. Thank you for the good questions and the conversation. Thank you to Regina for giving us an excellent talk. Thank you to the Crate Place and our servers for hosting us tonight. Hopefully you get a chance to thank them on your way out. Again, I'm Monica and I'm from the Women in Science and Engineering at UC Santa Cruz. We have these hats that I've been told to publicize. If anyone would like a WISE hat, I don't know, how much are they? They're $12. So if you would like to support us in all of our activities, we don't just do Science on Tap, we do a slew of outreach activities in local middle schools and high schools and elementary schools, as well as a lot of campus programs that try and, again, promote women and diversity in the sciences. You can support us by buying a hat. Otherwise, our next Science on Tap will be, again, the last Wednesday of the month. So it'll be April 25th. And if you're not on our mailing list, you can come and find me and I can get you on our mailing list so that you receive updates on the topics for the Science on Taps. If you have a suggestion for a speaker, please let me know. And then I also have one little plug for those. I'm going to bring Anna over here to talk about something a little bit political.
Okay, sorry everyone. My name's Anna. I'll keep this super brief. I'm a grad student in the Environmental Studies Department at UCSC, and I'm helping to circulate a petition that would put a renter protection ballot initiative on the ballot in November. So if we get enough signatures, this will go on the November ballot. We as a community will vote on it, and if it goes into effect, it will include a rent stabilization component as well as a just cause evictions protection for all renters in Santa Cruz. So if you are a Santa Cruz registered voter, you can talk to me and send a petition after, and I'm happy to answer any other questions after. I don't want to take up too much of your time, so thanks everyone for your attention, and thanks Monica and Regina also.
Thank you.
Rural New Hampshire.
The mic is still on. We wanted to make another announcement about an event happening in April you might find interesting. It will be a, quote, Earth Night event on Wednesday, April 18th, focusing on the effect of lighting at night and how that infects our environment. It's especially relevant to endangered species here in Santa Cruz and Monterey County. We will have speakers on the biological effects, also an astronomer. There's a fellow coming from the Zoological Lighting Institute who will be sharing a documentary on how lighting has affected endangered fireflies. And we would especially like to engage biologists in this issue because, as Monica was describing today, a lot of small animals are very affected by their environment, thermal environment.